Okay, good afternoon, everyone, and welcome to the Live Lab here at McMaster University. I'd like to acknowledge the land on which we are currently sitting is on the traditional territories of the Mississauga and Haudenosaunee nations, and within the lands protected by the Dish with One Spoon Wampum Agreement. It's important to note that teachings from the First Peoples of Canada have had an enormous impact on all areas of academic pursuits, from science to engineering to humanities. And without Indigenous knowledge, much of our current research would not be possible. My name is Joe Kim, and I'm the Academic Director of the McCall-McBain Postdoctoral Fellows Teaching and Leadership Program. This is a unique program dedicated to creating professional development opportunities for postdoctoral scholars at McMaster, the surrounding regional universities, and across Canada. And one of our fundamental goals is to provide early career researchers with tools to improve their knowledge translation skills and to be able to convey their research impact, which is exactly what we're going to do today with our 5-minute research talks. Okay, so take a look around you. You are currently seated in one of the most unique research centers and performance spaces in Canada. It's called the Live Lab. It's the large interactive virtual environment lab. It's a 106-seat research performance hall designed to investigate the experience of music, dance, multimedia, and human interactions. Lots of amazing features. So when we're using this as a research facility, there could be a musical performance, a lecture, some sort of performance. 40 of these seats can be wired so that we can measure EEG, brain waves, from 40 different people at once while they're interacting and watching a performance. Even the acoustics in this room, it's a virtual acoustic. So the actual room is very, very quiet. It's only 10 decibels loud, but I could make adjustments to this room. For example, I'm gonna just show you a very simple one here. This is a pretty good demonstration of the virtual acoustics of, this is just a fly moving around this room. But the room itself, if I clap my hands, that's the echo that you hear. I could just make a change and I could make this room bigger. And I could change the acoustics to my choosing. If we happen to be doing a study on attention, hearing aids, what's it like being in a loud, busy environment, we could change the stage. We could make this into a restaurant. We could have tables around. We could completely change it. We could measure from the audience things like EEG, galvanic skin response. I'm going to actually change this back to a smaller room setting. We could measure motion capture, anything that you could think of. So as a researcher and educator, for me, it's absolutely fascinating to use this facility. And today we are hosting the 5 MRET talks. So I'm very confident that you're going to enjoy today's presentations. And we have a lot going on in the world today. And we need the next generation of thought leaders to move us forward. So I'm going to pass things on now to Katie George to introduce our judges.
Thank you, Joe. My name is Katie George, and I am the Program Manager for the McCall-McBain Postdoctoral Fellows Teaching and Leadership Program here at McMaster University. Pardon me. And I want to welcome everybody for coming to what promises to be a truly amazing event. I've seen some of these postdocs speak, I've seen the slides, and I am very excited for today. I truly believe that this is going to be the best year yet because of all of the postdocs in the front here. So looking at our schedule today, we do have a couple of breaks sort of embedded, but I will say if anybody does need to use the washrooms intermittently, please do. They're just outside the doors and then the little hallway to the right there. You'll see the washrooms there. If anybody requires them, please, at any time, please go. So today, we're going to have the privilege of hearing 25 speakers. I will say on your schedule, One of the speakers will not be participating, and that is, I wish I had the schedule in front of me, in the afternoon. So I will let you know after the break which speaker will not be here today, just so that you can sort of cross it off in your mind as well, okay? So these 25 elite researchers from institutions across Ontario. So this is not just McMaster University being represented here today, and you're going to see that within our judging panel and within all of the speakers as well. The folks are going to be competing for the top 10 spots in our event next week, which is the 5 MRT finals. And unbeknownst to some of you, I think, there is a cash prize. So if you are competing for a spot in the top 10, you will win a cash prize next week, potentially, if you get the top spot. So not that there's any more pressure added to today, because everyone's going to be great, but have that in the back of your mind. Today, our speakers were tasked with translating what they do. Their very intense, innovative, creative research, and taking it all and shrinking it down into 5 minutes, a 5-minute talk. That is no easy task. And just to layer on top of that, the talk has to be accessible to the community and to other disciplines. Again, not easy. But I know that every single speaker is going to be able to deliver today. I'm quite confident. I want to thank all of the researchers with us today for their hard work in preparing these presentations. Their innovation, impactful research, and dedication to their field is going to inspire all of you. And I know that networking and connections are going to happen amongst this group. They always do every single year. There's a collaboration of some kind I hear about. So I can't wait to hear about this year's. Now, looking ahead, I wanted to introduce our judging panel, just so folks know where the judges are in the room. So if you're speaking today, turn around and take a little bit of a look as to who will be on our judging panel today. All right, so judges, as I call your name, please stand to be acknowledged. First is Katrina Carbonera. Put the next slide, Katie. Yes. C. Thank you, Paulina. So Katrina Carbonera, thank you, and you can sit. She comes to us from Brock University, and Dr. Carbonera is the postdoctoral affairs coordinator at Brock. She completed her PhD in 2025, where her research examined how adverse childhood experiences were associated with increased cardiovascular disease risk later in life. Currently, she works with early career researchers and assists them to bring their cutting-edge research to the world stage. Next, we have Catherine Mabry. I feel like there should be applause for you. Catherine is from right here at McMaster University, and Dr. Mabry is the coordinator of postdoctoral affairs and research training. She's also the vice president of the Graduate and Postdoc Development Network, or the GPDN. With over 15 years of experience in higher education, Pauline's going to yell at me, sorry, click. She leads collaborative programming, delivers research training, and supports career transitions for graduate students and postdocs. All the way from TMU, Ketan, we have Ketan Marbali, and Dr. Marbali is a postdoctoral and graduate student professional development specialist and 3-minute thesis coach at TMU's Faculty of Engineering and Architectural Sciences. A former research scientist with a PhD in cellular and structural biology, he combines deep academic training with career development expertise to support grad students and postdocs with their career and professional development. He has published several articles in University Affairs and Inside Higher Ed, served as co-chair for the GPDN Professional Development Committee, and is one of the co-leads on the multi-institution from postdoc to research faculty crash course. And last but certainly not least, I bring you Thuyen No. Here she is. Golf clap. From the University of Guelph. Dr. Nyon currently works as an educational developer in the Office of Teaching and Learning, where she partners with educators to strengthen student-centered approaches to teaching, assessment, and program evaluation. With a background in psychology and educational research, she is especially interested in how complex ideas can be shared in ways that are clear, meaningful, and engaging for diverse audiences. Thuy-Ann's research and professional work are grounded in equity, inclusion, and social accountability, and she is passionate about making research matter beyond the academy. So thank you to all of our judges for coming today. And without further delay, I bring you your first speaker.
Hello, everyone. Welcome to this event. Thank you, Joe and Kete, for the warm introduction. My name is Abrha Gebrehiwot. I am from McMaster University. My talk today will be on how different contraceptives reshape vaginal lipids and vaginal health. My research focuses on the interplay between metabolomics, microbiome, pathogen, and host cell interaction at the vaginal microenvironment to deeply understand the molecular insights influencing women's susceptibility to HIV and other infections. This vaginal microenvironment is rich of diverse lipids that have essential role in pathogen entry modulation, in maintaining epithelial layer integration, and modulating the immune and inflammatory responses in the vaginal microenvironment, all of which have great impact in influencing women and fetal health. A previous study from our lab pointed out that one of the mostly used injectable contraceptives, that is called the depot methoxyprogesterone associate, or the injectable shot, could cause a dysbiotic vaginal microbiota, which is characterized by highly diverse pathogenic microbe in the vagina, low estrogen, low lactic acid, and unusual high pH, as well as low glycogen, and also a thinner and leaky mucosal and the epithelial layer of the vaginal environment, all of which will increase the woman's susceptibility to sexually transmitted infections. This hormone-induced microbiome shifting lead us to the question, how does contraceptives reshape vaginal lipid metabolism? With this, we designed a lipidomics study employing Kenyan sex workers that were grouped into pill or the oral contraceptive taking, the control group or no hormone taking, and the injectable shot or DMPA taking. From this, we used 20 microliter cervicovaginal lavage samples for lipidomic extraction and then mass spec-based lipidomic analysis. Upon necessary data processing and bioinformatics analysis, we finally found about 1,000 lipid species. Interestingly, these hormonal contraceptives clearly classified the study groups based on their lipidomic signatures, as shown in the PCA result. And more specifically, the DMPA or shot-taking groups, when compared with the pill and the normal, the no-hormone groups, could significantly downregulate the phospholipids, sphingolipids, glycosphingolipids, while drastically increasing the storage lipids, triglyceride. All this lipidomic shifting in the vaginal microenvironment favors inflammatory condition, whereas the pill or OCP group could have a reciprocal impact, implying its anti-inflammatory effect. In a more in-depth analysis, the pill and the shot reciprocally or divergently impacted specific lipids that have signaling and structural role. Specifically, the ceramide, the sphingomyelin, glycosphingolipids, and fatty acids, where significant lipidomic dysregulation was previously associated with preterm birth. Another important point is most of these differentially expressed lipids were mainly derived from microbes around the cervicovaginal environment, implying an important interplay between the vaginal lipidome and microbiome. further lipidomic ontology enrichment analysis confirmed that the top-ranked differentially expressed lipids were mainly the storage lipids, the signaling, structural, and fatty acids. In summary, the DMPA showed imparted unique signatures of vaginal lipidome with down signaling, down regulating the signaling structural as well as upregulating the storage lipids. This all favors the immunosuppression or immune escape of the pathogens followed by chronic inflammation. And this will in turn lead to the increased susceptibility of women for HIV, STI-related and STI-related infections. At the future work, we will do functional pathway analysis, microbiome versus lipid integration, and targeted lipidomics on the top-ranked lipid molecules to verify these findings. With this, I would like to thank my supervisors, Dr. Char Kaushik, Dr. David Waldai, and Dr. Maisa for collaborating in the bioinformatics work, as well as our funders and all members of the Kaushik Lab and the McCue program for coordinating such a wonderful event. Thank you for your attention.
Hello, everyone. My name is Mohammadamin Ahmadfard. I'm postdoc at University TMU. And today I want to talk about permafrost thawing. Imagine a cold winter day way up in northern Canada. The weather is so cold that it hurts to breathe. Snow crunches under your boots. And still, hidden warming is happening under the surface of the ground. This hidden warming is destabilizing the ground that people are living on. And after some time, it can make the buildings get tilted or even fall down. So today, I want to talk about this threat and also an engineering solution being designed to fight against it. So I would like to first start to talk about permafrost. Permafrost is a frozen ground that acts like a concrete. On its top, there is a layer which is called active layer. This layer melts in summer and then refreezes in winter. But the real strength of permafrost is coming from its lower layer that acts like a concrete, acts like a foundation. So it holds up everything from houses and buildings and roads and airstrip runways. So due to global warming, this layer is warming up. So this ground has been frozen for decades and even centuries, and now due to global warming, it's warming about 3 times faster than the rest of the planet. So what happens inside it is that the ice that is mixed in the soil, it starts to melt. And so this soil gets softer, and so it starts to sink, shift, and buckle. And it causes that the buildings of northern communities get tilted and they fall down. So one thing that we should notice here is that this doesn't happen in just one hot summer. Actually, permafrost thawing has a long-term heat balance problem. So you can think of it as a bank account for heat. In the past, long, brutal winters were pulling out all the heat from the ground and keep it stable. But now, due to global warming, more heat goes into the ground that will be able to come out. So why? Because winters have become shorter, much snowier. Snow acts like a blanket, and it insulates the ground, and it prevents it that the ground loses its heat. So the other reason for that is that our buildings are releasing heat into the ground. Maybe these heat sources seem to be minor and negligible, but by time, over a few decades, so their heat effects would add up. Engineers, traditionally, what they do, they were using passive solutions. So like raising buildings on piles or stilts that let the cold air circulate underneath of the buildings, or using thermosiphons that act like chimneys. They extract heat from the ground during the winter cold days. But those long cold winter days are finished now. And now we should think of more active solutions. That's where geopiles come in. Geopiles resemble like our ordinary foundation piles that we need to use them anyways to use our buildings. And, but they have a difference. They have fluid loop inside them that gets connected to the heat pump that is in our buildings. And so what they can do, they can extract heat from the ground and use it for warming up our buildings. But it needs very precise modeling tools, because there is a very tiny temperature zone that is about minus or plus 0.3 degrees C that needs to be respected. So what we do in our group, we just design such systems to simulate the performance of geopiles, and the results are clear. Geopiles act better than when we don't have them. The permafrost thawing is much shallower. It has lower temperatures. But this project is not only about designing of buildings. It's about designing the whole northern communities, like roads, fuel tanks, airstrip runways. And it's a win-win scenario for the northern communities, like indigenous people that are on the front line of permafrost thawing. So the question that we now we need to ask ourselves is that, do we still continue building our buildings by ignoring permafrost thawing, or we switch to much more smarter ways like geopile that are now we have it in hand? Thank you.
Hello everyone, I'm Sathya. It's great to be here today. Water. Water is life. However, recently, freshwater lakes are changing in a quiet way, that they are getting saltier. By the end of my talk, you will understand why this matters to all of us, as well as our drinking water safety. Only 3% of Earth's water is freshwater. Out of that, only 0.3% is surface freshwater. Yet, it supports drinking water, food production, ecosystems, a big service. When there are salts dissolved in water, we call it saline water. There can be many salts like sodium, chloride, many more. And we know of several saline zones based on the dissolved salt levels in that water, starting from lowest freshwater drinking water to highest hypersaline ocean water. Have you ever seen water looking like this? Thick green mats floating on the surface. So these are algal blooms. Algae is a simple plant-like organisms living in water. They can produce their own food too, like using sunlight. Algal blooms are more common than you might think. As an example, just look at the top left side. Algal bloom from Finger Lakes, New York, and top right from home, Hamilton Harbor. Bottom left from South Korea. Bottom right, satellite image. In fact, algal blooms are everywhere. So NASA mapped these algal blooms. Just look at these tiny green dots all over the globe. Indeed, they are everywhere. So algal blooms are not always harmful, but when they do, they produce toxins or flavors and a lot of other harmful effects. Harmful algal blooms damage ecosystems, water resources, and local communities. You can observe this damage through these newspaper clippings all over the world. So what's really causing the harm? Cyanobacteria is the one causing the harm. So these are called blue-green algae, a special type of algae. They produce cyanobacterial toxins, hence cyanotoxins, which are toxic to humans, animals, pets, and wildlife. A common culprit is microcystis, forming larger blooms, affecting drinking water and lake health, so producing microcystine, a toxin that damages liver. So now we know about harmful algal blooms and their bad effects. These blooms are often associated with nutrients like phosphorus. That's important too. But there's one stressor that is not given good attention, that is salt. So this is the gap we are trying to understand and fill in this study. freshwater lakes are getting saltier due to road soils, agricultural runoff, and climate change as a major factor. So here you can see the saline lakes being studied and algal blooms in aerial green color. During our field observations, we understood and we observed that specific dominant cyanobacterial species in each lake varied depending on the salt concentration in that lake. Also, we found that some cyanobacteria produce higher levels of toxins at specific salinity threshold, which you will understand further in this slide, where 1,000 and 10,000 salinity levels produce higher toxins. Based on our field observations, we started a lab-based experiment to identify and quantify salinity effects of various cyanobacterial species and dig deep into their genes to understand why they behave so. So, what are we trying to address here? We are conducting an in-depth analysis to identify the intricacies between cyanobacterial blooms, toxins, and salt levels to shed light into less understood but much critical factor affecting us all. To recap, freshwater is essential and algal blooms are rising, but salt can silently turn them into toxic. Therefore, we identified salinity tipping points that put drinking water at risk. Thank you.
My name is Shardul Tiwari, and I'm a social scientist based out of University of Toronto. And my talk today is about our politicians' take on technology. And it is our politicians because I'm talking about Canadian context. So without further ado, let's take a look at the technology. And the technology that I'm talking about is carbon capture, conversion, utilization, and storage, CCUS, is a technology mix where you can capture carbon dioxide from air, or you can capture it from industrial emissions like steel and aluminum manufacturing that we see here in Hamilton. You transport it and you can store it underground. Or you can transport it and convert it using electrocatalysis as a process by separating carbon and oxygen atoms in CO2, combining it with hydrogen, and produce hydrocarbons and its derivatives. Fuels and chemicals that we can use in construction sector, chemical manufacturing, and fertilizers. But why am I a social scientist talking about technology? Isn't social science about human geography, economics, sociology, criminology, law? You're absolutely right, it is. But I am an interdisciplinary social scientist. I look at the intersection of technology and policy and actors because technologies are not developed in policy vacuum. They're developed by actors. And those actors in my research today are the political actors, both at the federal level and at the provincial level, because these are the actors who make policies about technologies such as CCUS. So how do I do it? I do it by looking at what is called as Hansards. Hansards are the political debates. So whenever our parliamentarians at federal level or at provincial level, they talk about technology or any other regulation that they want to bring in, or the legislation, they debate that in the parliament. And that debate gets recorded and transcribed, and it's called Hansard. So I study these debates to understand where these debates are happening, who are the actors, who are the political actors who are talking about it, and what are they saying about this technology. So let's take a look, a sneak peek into what I'm finding. The first is where. It's not everywhere where the debate of CCUS is happening. It's these provinces, British Columbia, Alberta, Saskatchewan, Manitoba, and Ontario, highlighted in yellow, where there are dedicated legislations to talk about carbon capture, utilization, and storage, along with the federal government. But in addition to that, like, what are the other interesting things that I'm finding? So the first thing I'm finding is that conservatives, they see CCUS as a central solution for them, for leadership, industry, and competitiveness, as seen in these two quotes. This act will further cement Alberta's leadership in this area, unquote. Quote, passage of this act will allow Alberta to exercise its leadership, endorsed by G8, G20, and the UN. But what happens when we go to the other end of the spectrum, the liberals? For them, it's one of the technocratic tools in the climate solution portfolio. It addresses a small aspect. And what does that look like? We should be out on the front edge by looking at international standards, we need the legal ability to actually approve CCS if science proves out. So a little bit of skeptical approach, yet supportive of it. So in the overall political spectrum, as we are seeing today in the federal government and these 5 provinces, almost everyone accedes to the need of CCUS in Canada. I think the world is looking at us. We are an energy superpower. We should be leaders in this technology. And when I say almost everyone, there are some exceptions to it, but these are our political actors and the politicians and policymakers. But the difference lies not if they want CCUS or if they don't want CCUS. The difference lies on how, what, and where. And this is what my next study looks like at the policy diffusion, that at different places where the legislation of CCUS is coming up in these 5 provinces through acts and legislation, how are these actors are learning from each other? How are they adopting from each other in terms of new development that are happening around CCUS? And that'll be my talk today. Thank you very much.
Imagine a Quebecois filmmaker who made a documentary about a local conflict in a fictional African country. And what can we say about this conflict? Actually, this film is called Sona Rape and Terror in the Heart of Darkness. It becomes a huge success, winning an Oscar. And this film is inspired by Joseph Conrad's classical book, Heart of Darkness, which famously strips African characters of any agency. So what we can say is that centuries later, a book still influences the way outside Africa people perceive the continent, and the way inside Africa people perceive themselves, contributing to the delegitimization of the emancipation movement that inspired the conflict. This example, taken from a novel by Blaise Ndala, a Canadian writer of Congolese descent, shows us that books shape identities, memories, and systems of power way beyond fiction. And this brings me to the paradox at the heart of my research, which is that as literary scholars, we know a lot about novels, and yet we know surprisingly little about the books inside of them. These fictional books, the ones that the characters read, love, debate, sell, and buy, are overlooked in the study of Canadian and French writers of African descent. And this is precisely this blind spot that my research addresses. So my premise is simple, is that the book is unlike any other object because it creates a recursive structure inside the novel that contains it, allowing it to reflect on its own conditions of production, of circulation, of interpretation, and legitimacy, all from within. So what do these fictional books reveal? Let me give you 3 examples. First, fictional books reveal the challenges faced by authors of African descent in the global literary marketplace. In other words, who gets to speak. In a novel by Fiston Mwanza Mujila, an author-historian writes a novel on Congolese history, but is pressured by a Swiss publisher to simplify and exoticize his manuscript to make it more marketable for a Western audience. What this illustrates is that actually very often our perceptions of Africa are still more based more on marketability than accuracy. Second, fictional books reveal the position of African intellectuals inside Western systems of knowledge. Who gets to be believed? In the novel by Samit Chak, a French writer of Togolese descent, a Malian academic publishes an essay called on terrorism and migration. And actually, it's a huge success in France. But she's heavily criticized for citing too many European authors. Now she faces this double bind. If she doesn't, her work is not perceived as scientific enough. But if she does, she's perceived as intellectually dominated. And this illustrates a real-world epistemic issue, which is that Western institutions are still the ones that decide what knowledge is considered legitimate. Now, my third example would be the way fictional books reveal truncated versions of history. Who gets to tell the past? In a novel by Henri Lopez, a French writer of Congolese descent, an historian writes a book on Congolese history. And more specifically in this case, it actually adds to it the role played by African soldiers in the First and Second World Wars, all the while enjoying one of Russia's most famous writers, Pushkin, especially a novel in which a young African boy, Pushkin's great-grandfather, is adopted by the Russian Tsar, Peter the Great. So what this shows is that if Europeans have shaped African history for centuries, the opposite is also true. Africans have shaped European history. And so what does this matter today? Because narratives shape the world. They shape public policies, they shape debates, they shape collective imagination in an era of misinformation, of confusion between reality and fiction, of information asymmetry. They reveal to us the way knowledge is created, is circulated, is marketed, legitimized, or delegitimized. A book never appears magically on the bookshelves. It follows a trajectory shaped by people, markets, and institutions. And by placing books inside the books, these writers invite us to follow this trajectory. Thank you.
So when you hear type 1 diabetes, muscle is probably not the first thing that comes to mind, but I'm hoping in 5 minutes it might at least be the second, or I'd even settle for the third. Type 1 diabetes is when the body becomes unable to produce insulin on its own. Insulin is the molecule that tells our tissues to open up their doors to get glucose in. This helps regulate blood sugar, and it also gives our tissues the energy they need to function. So if we don't have insulin, we cannot regulate our blood glucose levels. Now, type 1 diabetes is treated by using lifelong insulin therapy. And while insulin therapy is incredibly effective, managing blood glucose is incredibly complex and often unpredictable. Your blood sugar can be impacted by what you've eaten, your stress levels, how much sleep you've gotten, and something as seemingly unrelated as a sunburn can actually impact your blood sugar. So balancing this all is incredibly hard. And even if you do a really, really good job of managing your blood glucose, it can still impact the whole body in really detrimental ways. One of the tissues that we're really interested in looking at the effects of type 1 diabetes on is skeletal muscle. Skeletal muscle makes up approximately 40% of the body, so it's a pretty huge tissue. It's also a massive site for glucose uptake and storage. Muscle is also what allows us to be strong and independent. It's why I'm standing up in front of you talking today. So it's a really important tissue for overall function. In our lab, we've actually found that type 1 diabetes has a pretty profound impact on muscle. In this graph here, we're showing that individuals with type 1 diabetes experience age-related loss of muscle mass at an accelerated rate. It's occurring almost 10 years sooner in individuals with type 1 diabetes. We've also found that in muscle from individuals with type 1 diabetes, following damage or injury, there is a slowed recovery. We think that exercise might be a tool at preventing some of these negative effects, because exercise helps promote strength and recovery. It can improve our mitochondrial quantity and quality, so improve the way that we actually produce and use energy. And it may actually help mediate some of those blood glucose fluctuations that we'll see as a result of type 1 diabetes. And in fact, Diabetes Canada actually recommends exercise for individuals with type 1 diabetes. But there's a pretty huge but. The exercise guidelines set out for individuals with type 1 diabetes are the exact same as what's set out for everyone else in Canada. And perhaps the bigger but, nobody has ever actually tested these guidelines in individuals with type 1 diabetes. So we have no idea if they're appropriate or sufficient. So that's exactly what we're doing. We are taking the exercise guidelines to determine if, at baseline, type 1 diabetes causes any changes to the muscle and overall metabolic health. And we want to see if the exercise guidelines can help restore any differences that may exist. The guidelines suggest 150 minutes of aerobic or cardiovascular activity per week and 2 strength training sessions. So we're having our participants follow these rules for 12 weeks using at-home exercise. Exactly what you would do if you saw these guidelines on the TV or in a pamphlet. We're tracking all of their activity using Garmin smartwatches so we can see what they're doing every day, how much activity they're getting, and if they're adhering to the rules that we've set out in the study. We're also tracking their blood glucose using continuous glucose monitors so we can see how that's impacted by exercise. And we're taking a whole bunch of measures both at the start and end of this exercise program to look at stuff like strength, aerobic fitness, and in our case, what we're really interested in, what is happening to the actual muscle itself. From these results, we hope to determine a few important answers. Is there a best type of exercise for individuals with type 1 diabetes? And quite frankly, do the guidelines cut it? We are looking to find our areas of greatest loss or gain so we can better target these in future studies. And we're also hoping to develop programs that support exercise across the lifespan for individuals with type 1 diabetes. Ultimately, we want to improve the quality and quantity of life for the over 300,000 individuals currently living with type 1 diabetes in Canada by keeping them as strong as possible for as long as possible. Thank you.
Medical imaging is a marvel. It is a window that lets us see what lies within without ever having to pick up a scalpel. We can look at the intricate architecture of the healthy human brain, like in this magnetic resonance image, or MRI. But more importantly, we can see when tragedy strikes, like in this computed tomography, or CT scan, identifying a life-altering stroke by the red arrow. The goal of my research is to use these marvelous windows, and not just understand the outcome of stroke, but also identify who is at risk and how can we prevent future complications. My name is Dr. Nicholas Simard, but you can call me Nick, and I work at the Image Analysis and Medicine Lab at TMU. We start our journey with MRI because it is the gold standard. MRI is incredible at defining tissue-tissue boundaries, making it easy to detect borders and edges between different parts of the brain, like in white matter or in gray matter. On top of that, MRI is a safe imaging modality. It uses non-ionizing radiation. So to collect these images, we use waves that are comparable to radio waves. So whether you're getting an MRI or listening to your favorite tune, the risk is the same. Using MRI precision, we're also able to find subtle stroke warning signs in things like intraplaque hemorrhage, or IPH, which manifests in MRI as tiny little white spots. But IPH, they're unstable collections of plaques that act like a ticking time bomb. When they go off, a stroke happens. In my most recent research study, I analyzed over 300 patients that are historically susceptible to stroke, and I found a striking IPH pattern. Men over the age of 70 years old are 5 times more likely to suffer a stroke within a 2-year span if they have a dominance of right side IPH. Discoveries like this can enable clinicians to step in with life-saving treatment for a high-risk group before tragedy occurs. But, reality check, MRI is a marathon. Although it provides a wealth of information, it takes at least 15 minutes to just get a baseline brain scan. On top of that, MRI is expensive. They require massive magnets, specialized hypercooling, and heavy infrastructure. For perspective, in Canada, we have 450 MRIs for our 40 million plus population. And in the context of stroke, every second counts. We don't have time to wait for beautiful pictures. We need answers now. And that's where CT takes the lead. CT is a sprint. It's fast, accessible, and can take a comprehensive brain scan in under a minute. It's particularly important in settings where we have to make split decisions very quickly, like in the emergency room. So think of CT as our first responder in identifying stroke risk. But there are also limitations. CT uses X-rays, an ionizing form of radiation, so we have to be mindful of harmful radiation dosage. On top of that, CT is really good at looking at the big picture, but it's not sensitive enough to detect tissue-tissue differences, making things like IPH go undetected. So, MRI, CT, which one is more favorable? Well, what if you didn't have to choose? And that is the next step in my research, multimodal medical image analysis. I'm developing a translator that's able to take standardized CT and MRI and have them speak the same language. By using machine learning, we're able to take CT data, map it directly to the high detailed MRI space, essentially teaching a computer to look for stroke with MRI level knowledge, but only having acquired a CT image. This is a game changer for stroke because now we can take a 1-minute scan, translate it, and identify those IPH ticking time bombs that were previously invisible. On top of that, this multimodal approach isn't limited to just stroke. We can apply this translator to things like Alzheimer's, Parkinson's, or multiple sclerosis, providing sophisticated research insights to the entire neuroscience community. So with my research, in the future, I want to enable patients to have the proper care so that we're able to prevent like MRI and treat like CT, so that everyone can be safe from stroke in the nick of time. Thank you.
I really want to express my appreciation to Katie and to all the people to help today's event. So thank you so much, and good to see some new faces and some familiar ones. Okay, so let's get started. Hello everyone, I am Weng, also known as Julian from TMU, Toronto Metropolitan University, and today I'm going to present our research. Okay, so the laser pen doesn't work. About nanobubbles, tiny agents driving huge impacts. So I am going to start with this simple equation: ultrasound plus nanobubbles equals to huge impacts in biomedical imaging. So I'm going to focus on the first topic, just for the vascular imaging for today, because of the time limit. Oh, okay, so let's talk about ultrasound first. So in general, there are 3 types of sound waves based on their frequency ranges from low to high. So we're going to focus on the right column, the ultrasound, with the frequencies above 20 kilohertz. And so this picture just shows you one example that we can use ultrasound probe to do, for example, heart imaging. Okay, and thank you to the last speaker. So I'm going to compare 3 traditional imaging modalities: CT, MRI, and ultrasound. I'm going to use a human kidney for this comparison. So CT, it can provide high spatial resolution. However, it's going to lead to the radiation. But you can see the vessels in the kidney, right, pretty clearly. For MRI, it can also provide high contrast imaging, and there is no radiation. However, the scan time is long and the cost is relatively high. And also it's quite noisy. And more importantly, it can lead to, it's not suitable for everyone, especially for the patients with metallic implants. So I am going to show you a short clip from the movie Final Destination to support my saying. So as you can see, this gentleman is a huge fan of body piercing, no judgment. However, it's going to lead to a disaster if you do MRI, okay? And ultrasound, there are a bunch of benefits of using ultrasound: real-time, cheap, safe, or no radiation, and quiet. However, why traditional ultrasound has been underestimated for a long time, it is because of its low contrast and image quality. As you can see from the image up there, we can see a structure of a kidney. However, you cannot see the vessels in detail. So that is why we're doing what we're doing now. So we're trying to combine with nanobubbles. So let's talk about nanobubbles. So they're teeny tiny. The mean diameter is around 250 nanometer. So to give you some ideas, I'm going to show you a visual comparison between a single red blood cell and a single nanobubble. So as you can see on the left, the donut shape represents the single red blood cell, and the yellow dot represents our nanobubbles. So they are much smaller than even a red blood cell. Because of their smaller sizes, they can easily enter the vasculature by blood circulation. And also they can produce enhanced ultrasound signals for us to image. So this is why we can do vascular imaging. So now I'm going to show you one of our representative case study for the preclinical studies on mice. So we have the visual sonics machine, the Vivo F2, our ultrasonic machine. And we have the ultrasound probe, and we're doing nanobubble injection by tail veins. And this is the schematic of the kidney. So the upper video just show you a traditional B mode imaging result. Again, it's pretty bad. You can see like a structure of a kidney, but you cannot see the details, right? And also because the animal is breathing, so you can see the breathing movement periodically. So the bottom video shows our results. So you can literally see it's like a Christmas tree lighting up. So we have more information about the vasculature. And also recently, we can further increase the spatial resolution so that you can see more microvessels in the kidney. And I think it's quite interesting to have a cute visual comparison between a vasculature tree in the kidney and a real tree. And also, by the way, the trees are so unique. It's called dragon blood trees. So when the trees are injured, it's going to bleed. You're going to see the red liquid coming out. Our research is not only about producing visually enjoyable results, but also can produce or can make a huge impacts. So here I'm going to show you a representative example for cardiovascular disease detection. So from the animation, as you can see, the one vessel is, is clogged. So by imaging nanobubbles, we can easily tell whether the vessels is normal or clogged. So that's pretty much about my talk, and thank you all for your attention.
Hello, everyone. I have a good question for you. How many persons here, by a show of hands, have used phones, computers, or any modern technology before? Okay, great. Pretty much everyone. Now, all of these modern technologies that we've all used at one point or the other in our lives are made from minerals that are mined from the ground. When we perform mining, we generate waste that we call tailings. Tailings we store in what we refer to as tailing dams. My name is Peter Adesina, and what I'd like to share with you today is my contribution to the delivery of resilient dams and communities by mitigating a failure mechanism that we refer to as liquefaction in tailing dams. In the last decade, we've had major tailing dams failures around the world, including here in Canada. In Canada, we had the incident in Alberta and the one in British Columbia. Outside of Canada, we had the Brumadinho Dam disaster in Brazil that killed about 250 people in one instance. Tailing dams failures around the world have killed 2,650 people, including 317,000 people displaced. We have about 500 tailing dams in Canada, almost half of them here in Ontario. If we think of the fact that Canada's mining activities will continue to increase because of the demand for rare earth minerals that we need for defense equipment manufacturing and also for the development of modern technologies, that means we could potentially have more of these failures in the future. Tailing dams failures are caused by different mechanisms, chief of which is liquefaction. So what is liquefaction? Imagine that you have a stable soil and you subject it to shaking. If you shake the soil well enough, it can begin to flow like a liquid. This is what we call liquefaction. What happens in the liquefaction scenario is that the soil goes from a state where the particles have contact to one where contacts are lost, and this is because of pore water pressure increase within the soil. So we can have earthquake-induced liquefaction, and in Canada we have about 4,000 earthquakes that occur per year, meaning that we have huge potential for earthquake-induced liquefaction in the country. We could also have a kind of liquefaction that we call static liquefaction. This is caused by the self-weight of a tailing dam. It is the one that killed over 250 people in one instance in Brazil. So the essence of my research is to prevent and mitigate liquefaction in tailing dams. And I do this by assessing the susceptibility of the dams, of tailings to liquefaction. This I do by performing a test that we call the cone penetration test. So what we do in the cone penetration test is that we push a cone within the soil and we measure parameters that give us an indication of the state of the soil in the dam. We can perform cone penetration test in situ, that is on the field, but it is expensive and difficult. It's the reason why we perform cone penetration test in a calibration chamber that we have within the lab, where we have control over influencing factors. So when you push a cone within a soil, you can measure the resistance of the soil to the push of the cone. And we call this the cone tip resistance. We then normalize this tip resistance and we'll plot it against a parameter that we call the state parameter, which essentially gives us an indication of whether our soil is loose or dense. So we end up with an empirical correlation that we use to determine whether a soil in a dam is susceptible to liquefaction or not. If the soil is loose, if it falls in this loose zone, we can make the judgment that the soil is susceptible to liquefaction. So with my research, we can assess liquefaction susceptibility in tailing dams, thereby preventing failures. This means that we can protect communities living downstream of tailing dams, thereby preventing their untimely deaths. And that means you and I can use our phones, our computers, all of our modern technologies with less blood on our hands. Thank you.
So my name is Faizan Salim. Thank you for all of you to coming here, right? And in order to honor your effort of coming here, today's talk, I'm gonna make it all about you, right? More specifically, how I'm using my research to help and protect you on a daily basis, okay? So the idea is to somehow bridge the gap between the DNA research being done and how it can be used for the practical applications to help a common person or the common public, right? Let's just start with something basic. So, water, right? So I'm going to ask you something and then I'm going to add something into it, right? How many of you know what bacteria are? Good, perfect, everyone is educated. So, and what I'm going to add into it is that about 90% of the testing, clinical testing, water testing is done by measuring the bacteria or targeting a specific bacteria, but the problem is that it takes about 24 hours for bacteria to grow. So for 24 hours, you're in the dark, you don't know what's going on, right? Water testing, clinical testing, whatever you can think of. Now, it is, for water testing, we use the fecal indicator bacteria, and it represents the gastrointestinal disorders, right? Problem is the 24 hours gap. You don't know anything about it, right? You don't know what the water quality is good. Now imagine you go to the beach, right? And you're asking the person in charge, hey, is the water quality good? Can I swim in it? And he's gonna tell me, you know, we collected the samples today, maybe come and ask me tomorrow. But tomorrow you would know automatically, because maybe you would spend most of your time in toilet having diarrhea, right? That's a clear sign that the water quality wasn't good. So that's the one problem. First problem that I'm trying to fix. Second problem is that it only specifically gives you information about the gastrointestinal disorders, maybe. But what about all the other stuff? Where is the contamination coming from? What about other bacteria? Maybe Cryptosporidium, protozoa, viruses? No, nothing like that. That's the second problem. So what I do is I use DNA to basically cover up these problems or to fix these. I use the DNA to make things faster. Instead of 24 hours, you can use the DNA-based technology to amplify and target the specific DNA of that bacteria that you're interested in, or virus or whatever, right? And get the same results within 3 hours instead of waiting for 24 hours, making it much faster, right? Second thing, if you want to look into more in detail, I'm not going to discuss any results today. As I said, it's all about you, what I'm doing for you, right? So you can look into the papers that are related to this particular research that I'm discussing right now. Secondly, the problem with the DNA-based methods nowadays is that sometimes it can be producing some errors. It's not foolproof. So what I do, I identify a specific DNA sequence or a part of DNA that is only present in the thing that you want to test for, right? And then develop a method that you can use basically to target that particular bacteria or pathogen without caring about like whether I'm going to get any false positives or negatives. Third thing is, as I said, the bacteria represent one thing and one thing only. What about all the other stuff that you want to measure? That's where the DNA sequencing comes into. I use the DNA sequencing specifically on the DNA that we collect from the water samples, and at the same time, instead of targeting one at a time, we identify everything. Viruses that are present in there, how many are there, and what they can do, and all the bacteria, all the protozoa, whatever you can think of, whatever that has a DNA in it, and that thing was present in the water, you can detect it, quantify it, and if you want to read more, you can look into the paper that I have in here. After that, you can test the water for as long as you want, right? But you cannot fix the problem if you don't know the root cause of it. Where is the contamination coming from, right? That's another use of the DNA-based technology, that you can identify that which particular organisms are producing that contamination, or which particular organisms whose fecal matter is being transported into the water that creates the contamination. If you want to read more into it, you can look into my paper. And through identifying the contamination sources, you can help in the remediation steps to protect the water sources for a longer period of time. And thank you very much for listening to me. And feel free to contact me if you have any questions, or maybe not.
Hi, I'm Kaia. I'm an evolutionary biologist from the University of Toronto. I study social evolution in mammals, and I'm going to tell you about my challenge starting large males. Most people I ask think that most mammals have larger males than females, and it makes sense. There are plenty of examples where this is the case, and it's taught formally in university courses. Oh, and it also goes along with evolutionary theory, because in mammals in particular, females are very busy. Not only are they often gestating, but they're also then lactating, and so they're reproductively unavailable a lot of the time. This should create a lot of competition for those rare receptive females, and should select for bigger, brawnier males. Darwin stated that most mammals have larger males in The Descent of Man, 1871, as a matter of common knowledge, and many others followed suit, citing Darwin. Darwin said so. Most mammals have larger males. The first to actually look at the evidence was Catherine Rawls in the 1970s, and she found that actually there was a lot of monomorphism, males and females of the same size, in mammals, especially those mammal groups with lots of species. So there are a couple of problems, as you may have intuited, with this larger males narrative. The first is that most mammal research is on large charismatic species, primates, carnivores, ungulates. So that's most of the data we have. But most mammals by far are rodents or bats. The second problem is that comparative studies across mammals have not so far used statistics to determine which species have males and females of the same size or different size. And that's just because the widely available data are averages. So they're stuck comparing average male mass to average female mass, but you can't use statistics to compare two numbers. So the determination of which species have males and females of different sizes has been quite arbitrary. So we decided to go back to the question, how often are males actually larger than females in mammals if you sample mammal groups according to the number of species they have, so lots more information on those rodents and bats, and if you use statistics for each species to determine are the males and the females same size or different sizes. We spent a long time going back to the primary literature, extracting averages and some measure of variance for the data on both males and females across mammals across the world. And we sampled each mammalian order and then each family within those orders according to the number of species they had. We only took data on wild adult individuals. We required at least 9 males and 9 females as a sample size for the data to be included. And we excluded pregnant females. And then we used statistics to determine for each species, are the males and females the same size or different sizes? And we found that while a big slice of the pie in mammals had larger males, that's the green slice, it was not most mammals. And actually almost just as many had males and females of the same size. And it turned out that it was really important, I don't know why this advances so quickly, to sample each group according to the number of species, because a full half of rodents had males and females of the same size, and a full half of bats had larger females. And that really changed the story. The study is important because people didn't just stop at saying most mammals have larger males. From the very beginning, Darwin said most mammals have larger males, and man is more courageous, pugnacious, energetic, inventive than woman. Trivers said most mammals have larger males, and they're also more aggressive. Maury said that, oh, this is all because they have more testosterone. This is all based on this fundamental belief of what was the norm in mammals. And it led to a lot of biological determinism on what was natural in mammals and therefore also in humans. So we thought it was important to correct the record. Thank you very much to my co-authors, funders, and media.
Hi everyone, my name is Andrea, and today I'm going to talk about the interaction between proteins. So first, I want you to imagine that you go home. How will you measure the length of a table that you have at home? Most of you will run to the toolbox and will pick a ruler and it will measure it. But have you ever think about how do we measure interaction at a nanometer scale level? Nanometers is 100,000 times smaller than the width of your own hair. So how can we say that two proteins interact and how far they are interacting between each other? So for understanding this, I want to go back in time, and I want to go to medieval times. Back in the day, what happens is that when a village was in trouble, they will go to the tallest towers in the village and they will light a fire on the hopes that the next city will see it and reply to it. If this fire was dim, or if this fire was not, if the city was really far away, what will happen is the city wouldn't be able to see that fire and it won't be able to reply to it. In biology, we don't have towels, we don't have castles, but we have biomolecules, and we can label them with fluorophores. So what happened is that we can have two molecules labeled with different fluorophores, and we can see how these two interact with each other. So the molecule that is sending the signal is called the donor, and the molecule that is actually receiving these signals, we are calling the acceptor. And this transfer of information between each other, we call it FRET, Foster Resonant Energy Transfer. So how this works? So normally we have a molecule that is going to be in a relaxing state that we are going to excite with a light. When this molecule traps the light, it's going to be excited, and it's going to get to the excited state. This energy, this molecule needs to lose it somehow. And there are different ways that it actually can happen. So one of the ways is releasing heat. Another will be motions. Another way will be fluorescence. But if it's lucky enough and it has a fluorophore next to it, what it can happen is that energy can transfer to this other molecule. In this case, this molecule will get excited and it will release light. This means that depending on the distance between these two molecules, we're going to see more or less signal from this molecule that we call acceptor. So if the two fluorophores are really close together, it's really easy the communication between each other. And we're going to see a really bright signal coming from the acceptor. As we separate them and they go far apart, what is going to happen is that this communication is going to get dim, it's not going to be as efficient, and we're going to lose signal from this acceptor. Thanks to a mathematical model that has been widely proved, we can actually, with the signal coming from these two molecules, determine how close these two molecules are. But the problem is that we can only work with distance that are actually within 10 nanometers. So what happens if we want to study something that is beyond those 10 nanometers? There's another system that we can work with it. In this case, the acceptor is not a fluorophore. In this case, the acceptor is a nanoparticles, a metallic nanoparticles. It's going to work as a quencher. So what is going to happen this time is that the fluorophore that is going to be in excited state is going to give all the energy to these nanoparticles, and we're not going to see no lights coming out. We're going to lose all the signal. In this case, if the two are together, no signal will be coming out. As we tear them apart, what is going to happen is that we're going to receive more and more signal from this donor. This system has a distance dependency way difference from the one from FRET, which is going to let us study system beyond those 10 nanometers. Also, there are some scientists that has reported that the distance in which we can work with this, it can depend on the size of the nanoparticles and the shape of the nanoparticles. So what is the problem here? The problem is that this interaction between the fluorophore and the nanoparticle is really complex, and there is no a consensus agreement on how these two interact. So there is no a mathematical model that actually can explain this interaction in a consistent way. If we don't have a mathematical model, what is going to happen is we cannot actually apply this to real samples. In our lab, some of our work that we have been published studied the interaction between a fluorophore called terbium and a metallic nanoparticle, which is gold nanoparticles. In this case, this work discarded some of the mathematical interactions that these two have, but still there is some information that needs to be done. So what we are going to do is develop a system that allows us to study different distance in a really controllable way to actually find the best fitting for a mathematical model and actually kind of be able to sense these molecules from far more distance. So that's what we're going to do. Thank you.
Well, hello, everyone. When I studied civil engineering, I always wanted to create an impact on the society. And I was lucky as my research gave me this opportunity. Let me take you with me on my journey from healing concrete to healing spaces. Before that, I'll introduce Mr. Cracky. Mr. Cracky was a happy, cheerful concrete structure until crack starts propagating into its core. In addition, Mr. Cracky was designed with inaccessible, narrow doorway, confusing layout, and steep stairs, until we, civil engineers, stepped in, and everything changed. So, civil engineers do not only work on constructing buildings and so on, but we also consider various aspects, from construction material, moving forward to designing inclusive and accessible built environment. And talking about construction material, let me zoom in on a familiar problem. Cracks. Cracks propagate into the core of concrete, affecting its performance and durability over time. So these cracks allow gases and liquids to penetrate through the concrete core, affecting its performance, and not only this, it's costing billions of dollars to repair every year. That is why we thought about something. This is a motivation for us to think about something else. So, inspired by nature, when your skin is cut, it heals itself, okay? This is how engineers reimagined concrete with similar abilities. And guess what? Concrete can self-heal. However, since it's man-made, it requires an extra help. And this is what we do in our lab at McMaster University. We provide this extra help by providing what is called self-healing capsules. These capsules are integrated within the concrete mixing process, and once the crack propagates into the core, they carry a healing agent, and this healing agent will be released to fill these cracks. However, despite the promising results of self-healing, many questions have been raised. And this is because concrete, capsules, and the interface between them each has their own challenges. And it's really hard to come up with a mixture that is compatible with each other. And this is what we do in our lab. We look at what's the optimum design and geometrical and mechanical properties for the capsule to be compatible with concrete. Additionally, we look at what if these capsules did not survive the harsh mixing conditions. And also, we look at how can we come up with a standardized method to test the efficiency of self-healing. So, by optimizing the capsule design and performance, we look at what are the best or optimum design conditions for the capsule that remain intact in the concrete, yet rupture upon cracking. It's important for the capsule to rupture to be able to heal this healing agent, because if it debonded, this healing agent will be considered a waste. And in addition, it's going to keep voids that will compromise the concrete strength. We also look at if the capsule is going to survive the harsh mixing conditions, because whether if it ruptured or squeezed and then ruptured, or it is being punched by the aggregate available through the concrete mixing process, it's rendered inefficient. And also, we look at developing a test in order to come up with what if this self-healing process is successful or not. And to do so, we look at standardized testing method, adopting what is called a shrinkage ring test in order to find out if this test can be universally accepted in order to evaluate the efficiency of the self-healing. If you're still wondering about the title, well, this is because I do much more, okay? And even in 5-minute talk, I want to highlight that I do much more by moving from healing concrete to healing spaces. However, this is for another talk. To check out our work on accessibility, please scan this barcode. Thank you.
Hi everyone, my name is Milena. I'm an ecotoxicologist, so I study how pollutants affect our environments. And right now, as part of my postdoctoral research, I'm working on an experiment that's probably going to be the biggest experiment I'll ever be part on. And that's what I want to talk about to you today. So it all starts here with every one of us. Every time you're using disinfectant wipes, fabric softener, or shampoo, you're releasing something called QACs, or quaternary ammonium compounds, into the world. These substances are basically the active ingredients of these products, and we're talking hundreds of products, billions of dollars in market value. They go down the drain, they enter our environments, and still we don't really know what happens once they end up there. And that's what we wanted to find out. And this is where it gets interesting. So this is a picture of us on one of our research lakes, and what we do in that exact moment is adding QACs to a pristine lake. And now you might be thinking, wait a minute, you're polluting a lake? Let me explain. So first of all, what are QACs? QACs is a big number of compounds. And to really explain to you how they work, let's focus on one example, benzalkonium chloride. It's also the QAC that is most often used and the one we use in our lake experiment. Benzalkonium chloride has a permanent positive charge at its nitrogen atom, and that's really the characteristic of QACs. This positive charge makes it attracted to the negative charge of the surface of microbial cells. And once it attaches, it punches holes into the membrane and ultimately the cell dies. And that's really how they clean. But there are two main issues with these substances. First of all, they're non-selective. So they don't just kill the bad guys. They also affect potentially beneficial bacteria. The second issue is what we see in our environment is they don't break down easily. They're basically persistent, which means once they enter the environments, they're affecting these long after they enter them. So our research question is simple. What happens if we add QACs to an environment and when we expose it to QAC concentrations that we also find in our rivers downstream of our wastewater treatment plants? So you might be thinking, well, why aren't you testing that in a lab? And a really important thing that we're missing is realism. In a real lake or a river, we have thousands of species interacting. We have temperature, we have sediments. It's just way too complex to test it in a lab. Now, we do have QACs already in our environments. Why aren't we looking at these systems? Well, this makes it really difficult to isolate the effect of QACs because these systems are already polluted with pesticides, with pharmaceuticals, with microplastics. This is why we're working in remote locations, so remote lakes at the IISD Experimental Lakes area. This is an area in Northern Ontario, and obviously this experiment is huge. It takes 2 years to expose the lake, and it includes lots of different groups who are working on this together. Some of them are focusing on where the QACs go in the systems, others look at fish, and it's basically a big, giant puzzle. Everyone has their own piece. So what's my piece of the puzzle? Me and my group, we are looking at microbiomes. So these are microbial communities that live either in the environment, so in sediments and biofilms, or are associated with organisms. So they live in fish or in insects. These are critical, both for the environment and the species. The species can't live without them, and the environment can't function without them. We use DNA techniques to track who's there, what are they doing, and how do they respond to QACs, and in the end, how does that affect the environment or the organisms? Together, the evidence that we're gathering will help us inform and get an idea how we have to regulate QACs and drive safer alternatives. Because right now, every time you reach for these disinfectant wipes, these compounds are affecting microbial communities and reshaping them. And our work will show us how they do that and will help us make informed decisions how we can clean our homes without compromising our waterways. Thank you.
Okay, my name is Dr. Ailsa Edward, and I'm from the University of Toronto. And I'm going to be talking to you today about turning trees into chemical sponges. We all know that climate change is a big issue that we're currently facing. I'm sure you're all aware that atmospheric CO2 levels are rising at an alarming rate. This is causing devastating environmental impacts. We are seeing increasing reports of wildfires, such as those in Diaspora last year, and those in the Palisades in Los Angeles this last year as well. These are having devastating consequences on people's homes, lives, and the environment itself. The issue with these wildfires is that they are causing a rising increase in CO2 levels, which causes a vicious cycle. As CO2 levels rise, our planet gets warmer. This causes more wildfires, which then increases the CO2 levels again. And we end up in this vicious cycle. But how can we stop this? A report by the World Meteorological Organization also reported that we are seeing triple the amount of CO2 since the 1960s. And this is increasing year by year. A solution to this problem is called carbon capture, which we already heard a little bit about before. What this does is we take CO2 from the atmosphere, and we can either store it underground until we need to use it later, or we can transform it into more useful products chemically. The problem with this is that we need a material which can effectively capture that CO2 at large scales. But the solution to this might not be as hard as you think. A simple structure that we all use daily, the sponge. A sponge is a porous material. It's made up of tiny holes which can uptake water. When we immerse this sponge in water, it soaks up into its pores. Then, when we want to release the water from the sponge, we squeeze it, and we have our water out. For these materials, even though they're so lightweight, we can think that they can take up so much water, and then they become heavier, and we can release them as well. This is where my research comes in. We look at a material called a moth. This is just a chemical sponge. It has pores, like a sponge, which can uptake things like CO2. When a moth is immersed in the atmosphere, it can hold up the CO2 inside its pores. Then, if we apply a gentle application, something like heat, we can then release the CO2 from the pores of the moth. These moths are really important in society, and they have just won the Nobel Prize last year. But these materials sound great, right? They can take up lots of CO2. In fact, per 1 gram of material, a moth can take up the amount of CO2 if laid flat as the size of a football field. But the issue comes in the synthesis of these materials. They often use harmful reagents. They take a long time to make. They need harsh conditions, things like high temperature, high pressure. And they sometimes use oil-derived resources, which defeats the point of trying to reduce CO2 emissions. So we need to think of solutions. And how we can do this is to look at using sustainable solvents, things like water. We can look at reducing the time it takes to make these moths. We need to make them at ambient temperature and ambient pressures. And we need to look at lowering the cost by doing all of this. A solution to doing this is to take biomass-derived resources to make our materials. The work that I do looks at taking lignin. This is the woody component of trees. It's a chemical that we can extract to make vanillin. Vanillin you might recognize in your everyday life as the essence that makes the taste of vanilla flavoring. So what we do is we derive a vanillin into a new structure. We add it with a metal, and we heat it, and we mix it at room temperature in ethanol, a sustainable solvent, and we end up with a moth. We can then test its CO2 uptake, and we see that it hits the target CO2 uptake for these sorts of materials. I was able to show that we can make a biomass-derived moths in just 5 minutes using sustainable synthesis conditions from biomass. Now, this technology is not just fantasy. We're actually using this in real life today. An example of a moth being used for carbon capture was reported for the first time here in Canada, called CAF20. Thank you.
Good afternoon, everyone. I want all of you to take pause for a moment and think about the delicious food that we had in our lunch. Statistically, half of the people that are sitting in this room are alive because of a single chemical discovery which happened 100 years ago. So, I am here to produce fertilizers and for a sustainable growth and to feed the world. So what is new about it? The equation nitrogen plus hydrogen giving ammonia changed the history of mankind because it is the greatest invention of the 20th century because it literally gave life to Mother Earth. Without this, our population would be half of the number what is there. However, every coin has two sides. It leads to humongous amount of CO2 emissions, and it constitutes 2% of total CO2 emissions produced annually in a year. So why? We have read this equation in our high school days and requires a temperature of 500 degrees centigrade with 300 atmosphere to take this to do this reaction. So why we can't fix it? The solution lies in the molecule itself, nitrogen with the triple bond. It is the nature's strongest bond. It is the nature's version of chemical superglue. So it is very difficult to break it. In order to break it, we need an instead of an heat source, we need an electricity source so that we can do this reaction at room temperature and pressure. However, in doing so, we produce hydrogen bubbles, the competing hydrogen evolution reaction. So we must make the process selective enough. So we chose the process of electrocatalysis. However, in any catalysis to be successful, it must be successful in three areas: stability, activity, recyclability, and selectivity. However, all the catalysts which have been developed dances up and down this diagonal line. So no one has able to hit the sweet spot, the most active zone. Also, we find here that in order to reduce hydrogen, it is much easier because it occurs at lower potential compared to nitrogen. Henceforth, hydrogen evolution reaction dominates over nitrogen. So we at the University of Toronto tried to make a synergistic relation by the terminology that I am weak but we are strong. In the catalyst, which are all metals, we tried to cover the metals by an organic layer and form this cobalt phthalocyanine complex. Why we do that? So we impede the hydrogen to enter into the metal sites and allow specifically nitrogen to enter. What happens? Because of this nitrogen molecules is adsorbed onto the surface, and by the nature of back bonding, which is like an electronic choreographic dance, we basically enter the nitrogen lone pair of electrons enter into the vacant d orbitals of the metals, and it pushes away into the lone pair into the pi orbitals, and therefore the nitrogen bond length increases, the bond order decreases, and the nitrogen molecule gets activated. Using this, we then move forward to how this occurs. We prepare a molecular like a molecular self-assembly and how this reaction proceeds. We use computational modeling technique where the nitrogen sits onto the metal sites, and after each hydrogen sequential addition, we find that after first hydrogen addition, it can go under two pathways. First, where the hydrogen is added to the distally outward nitrogen, henceforth the terminology distal pathway, or the hydrogen can get added to the alternating nitrogen. It's called the alternating pathway. So these two probabilistic pathways are there, which results in ammonia formation. Thermodynamically, alternating pathway is more preferred over distal pathway for this catalyst. However, we only do not make the molecules dance in our computer screen. We have translated the technology, upscaled it, and made it into a small prototype, and we have climbed the technology readiness level, and we are in the TRL scale of 5. And thank you, everyone. And I would like you to encourage for the electrifying the future for food security.
Good afternoon, everyone. Thank you for being here today. My name is Sayana Pereira, and I am a postdoctoral researcher in the Chemistry Department at McMaster University. The title of my talk today is From Powering Our Life to Close the Loop, where I'm going to discuss why lithium-ion battery recycling is essential for a sustainable future. Before I start my talk, I'm going to ask more or less the same question that Peter asked. How many of you here has one of these devices? Can you raise your hands, please? Yeah, I think everyone. And all these devices, they have one thing in common. They are powered by lithium-ion batteries. Lithium-ion batteries change how we live. They are power our phones, or cars, and also help store renewable energy. The lithium demand is increasing fast over the years, especially because of the electrical vehicles. However, this is great news when you think about decarbonization, right? But this also brings new challenges, such as the battery waste and also the limited source. The battery waste, as the demand of lipids increase, the waste also increase, especially here in Canada, where the cold weather stresses the battery, which help the degradation of the battery go faster. Also, lithium is a limited source, where Canada holds around 4 to 5% of the lithium resource in the world. That this can be sounds a lot, but when you think about the demand in the future, maybe it's not gonna be enough for whatever we needed. So what is inside of the battery that can be recycled? A lithium-ion battery works by moving lithium ions between two electrodes. The anode that usually is a graphite to the cathode that can offer different chemistry, such as LFP and NMC. Every time that you charge or discharge the battery, you change the chemistry inside a little bit. So over the time, the performance of the battery is gonna drop, and the battery is gonna reach the end of its life. But this doesn't mean the battery is empty. The battery still have valuable lithium inside and also critical metals. The challenge here is how we can recover them efficiently and also environment friendly. So the recycling starts, now we're going to introduce one of the methods that we can use to recycle the lithium-ion batteries. Lithium recycle starts with the collection and transportation. After that, we need to separate the batteries based on the chemistry. Usually we have two groups. One is based on the LFP materials, and the other one, the NMC materials. This step can be done manually or with automated system. After that, we can disassemble the battery. We have two options. We can disassemble the battery, which is gonna give us all the components separate, and then we can recycle them directly, or we can crush them and obtain what we call a black mass. For the talk today, I'm gonna focus on the black mass. The black mass is treated by using hydrometallurgical process, which is a mix of the solution that contains sulfuric acid with hydrogen peroxide in order to dissolve all the metals there. After that, we're gonna obtain two products. The first one is a lithium solution, which contains all the metal dissolved there, and also a solid phase that we call a recycled graphite. The lithium solution, we can play with the pH, and with that we can precipitate all the metals that we have there. But for the talk today, I'm gonna focus on the recycled graphite. The recycled graphite, we need to evaluate how pure it is, so we can use different techniques for that, such as powder XRD to identify any crystalline impurities that we have there, also TGA, SEM, and solid state NMR. As a result, we got a graphite that was between 95 and 99% of purity, and also we could identify that the binder is still intact after the whole process. And finally, you could test the recycled graphite in a real battery, and we were able to obtain 95% of the theoretic capacity, which is an excellent result. With that, I was able to close the loop of lithium-ion battery recycling and turn the waste into a valuable resource. Thank you for your attention.
Hello, everyone. Lovely to see you all. Okay. I started my research career by studying the ecology of this super cute little caterpillar in the high Arctic. And now I'm an economist. Yet I'm still a biologist. The reason I say I'm still a biologist is because economics is biology. Now you might be thinking, what is this English guy saying? How can economics be biology, right? Well, for the rest of my talk, I'm going to explain why economics is biology, how this idea relates to my research, and the implications of this idea. Let's start with some fundamental properties of biology. The first is evolution. Humans have evolved over millions of years. All other living species now have similarly evolved from other species now extinct. For the passing down of information about previous selective pressures, biology needs some form of information storage. DNA serves that purpose by storing the instructions to build each individual and by being passed from parent to offspring. Two central species interactions are competition and mutualism. We see competition between and within species over resources, territories, and mates. We also see mutualism between species and within species. A classic example is bees pollinating flowers while also collecting nectar for their hives. Let's look at economics. Our economic systems are constantly evolving at all scales. I didn't grow up with a dial phone, but I think some of you here might have. And now we have high-end computers in our pockets. We also have hereditary information in economic systems. Laws are written down and passed from generation to generation, evolving over time. Finally, competitive and mutualistic interactions have profound economic impacts on economics. Companies and countries compete over market share, resources, and territories. But humans also interact mutualistically, with national health services being a salient example. Now let's connect this idea to my research. I examine energy flows in biology and economics. And the main overview of this research and many other people's research is that energy is critical to biological and economic systems. Both systems intake energy and then process that energy. Species intake energy in the form of other species or sunlight, and then allocate that energy with matter into biomass, movement, survival, and reproduction. Economic systems also intake energy in the form of oil, solar power, and food. This energy is then allocated into production, infrastructure, and many other purposes. That intake of energy and processing is then converted into growth. Populations increase in nature through continued energy intake. We also know that higher energy intake and higher energy efficiency directly grows our economic output. Okay, energy is critical to biological and economic systems. So what? How does this idea help us? Well, over the next century, we are going to overshoot several ecological ceilings, including climate change and biodiversity loss. But we also need to ensure that all people are healthy, educated, and have access to dignified jobs. Managing our human and our natural systems to be between the ecological foundation and the ecological ceiling and the social foundation is the profound question of our time. My personal route through this question is energy. We can use the core knowledge that we have gained from understanding energy flows in biological systems to inform how we design our economic systems. So you see, I am still a biologist. I will end with a quote from a T.S. Eliot poem. "We shall not cease from exploration, and the end of all exploring will be to arrive where we started and know the place for the first time." Thank you very much.
So all of us in this room know that the climate is changing with dangerous and disastrous consequences. A problem of this magnitude requires a big change. And to change everything, we need everyone. And yet, there is a lot of climate skepticism, denial, and really, disinformation seems the only thing changing and growing more than climate change itself. At the same time, there are a growing number of climate data. However, despite they are very accurate, there is a problem in communicating this data, and having more data does not imply more awareness. And at the same time, we have also another issue. The people that are the most affected by climate change are also the ones that are much more removed from the debate around climate change. There is a lot of inequalities we are facing, right, when we talk about climate change? And all of this to say that a lot of policy and climate policies have been centered more and more in type of solutions that are detached from the needs of the most underserved communities. All of this topic, misinformation, climate, equity, justice, is exactly what we do. So I'm Elisa Privitera, and I'm a postdoctoral researcher at the University of Toronto, and today I want to walk you through my path of research, and especially a project I've been taking part of in the last years that precisely tackle the problem of climate, environmental injustice, and misinformation. This project is called Just Transition in Action, and is a community-based research project that is focused on understanding what it takes for communities, especially the most underserved one, to flourish in the low-carbon transition. It's precisely focused in Scarborough, that is notoriously a more underserved and low-income communities in Toronto. So, within this project, we conduct all a series of listening sessions. Listening sessions are a series of action-oriented conversations that seek to explore how climate action intersect with people everyday concern. You can see here a series of activities, meeting multiple people, more art-based, workshop, community mapping, and all of this. And let's focus a bit on the process. What we did is that during 2023, we map community needs, community priorities, community visions for the future for their own neighborhood. And then the year after, we did a second round listening session where we identify those climate actions that were actually trying to address those needs, community visions that we mapped the year before. By doing so, we had a taxonomy of climate action that were at the same time mitigating climate change effect, also adapting to climate change effect. But at the same time, we're addressing equity justice, while we were also building collective capacity. We were talking with people, we were having discussion with people, conversation. And all of that, on top of this, all the process was through what I call a community-based review process. So we were involving people in double check if the data we collected were understood properly, and also by doing so, we were also sparking additional conversation, sparking additional awareness around these topics. So let's go toward the takeaways. I'm mindful of the 5 minutes. So first takeaways, we use community priorities as an entry point to talk about climate change and climate effects in daily life. By doing so, we really were able to understand, define all a series of common actions that were much more aligned with local needs and needs of people that are the most underserved people in our communities. And by doing so also, we expanded our understanding of climate action. Climate action are not anymore only technology solution, but are, first of all, are a problem of daily life struggle. Affordable housing is a climate action. Public mobility is a climate action, right? And maybe most importantly, by doing so, we did create space for rethinking, resisting, reimagining a different future that is more green and sustainable. So in this way, the solution we found were climate solution for all. I want to say and finish with some key aspect that is also the way we produce this knowledge. There is a fascinating strand of knowledge within social science, like post-normal science, disseminatory knowledge production, participatory action research, that already talk about the importance of extended peer community, involving community, right, in producing knowledge. However, very little knowledge has been produced on how working with community and not only studying community can be good for producing common action that are just action. And let me finish. By doing so, we really have a science that is much closer to people, more intelligible, more aware of real needs of people, more just, more transformative. And if we really, to change everything, we need everyone, this means that we need also scientists that are able to experiment with more inclusive and just way to produce and disseminate scientific knowledge. Thank you for listening to me.
92% of promising drugs fail when tested in humans. It's a crisis that costs billions of dollars and delays life-saving cures. My name is Sabin Saqib, and today I'll tell you how my research aims to solve that problem. So we've all seen headlines like this: miracle Alzheimer's cures, cancer breakthrough. Just earlier this week, there was a lot of buzz about a cure for pancreatic cancers by Spanish researchers. And these headlines give us a moment of real hope. But then the news cycle moves on and the promising cure vanishes. What happened? Well, to understand that, we have to understand the drug development process. So it starts with identification of chemicals that might produce a therapeutic effect. And then we do testing in animal models. We test to see if the therapeutic effects appear in lab animals such as mice or rats. And if we see positive results from that, we move on to testing in humans in clinical trials. And here, again, we're looking to see if the therapeutic effects are looking good in humans and to see if there's any adverse effects. If all goes well, we move on to regulatory approval and bringing the drug to market. But the flaw with this is that lab animals, mice and rats, are not humans, and you're not getting human relevant data to begin with. So when you make the transition from the second stage into clinical trials, a lot of the drugs that seem promising are no longer producing the desired effect. So to solve this, researchers have been looking at alternative to animal models for a long time to get that human relevant data from the early onset. And one of the most promising alternatives that have come out is a technology called organ-on-a-chips. So this technology, you take human-derived cells and coax them into forming tissue-like structures and organ structures outside the human body. So you might have brain-on-a-chips that react to stimuli. You might have heart-on-a-chips that pump nutrients. You might have kidney-on-a-chips that filter waste, lungs-on-a-chip that perform gas exchange, liver-on-a-chips that break down drugs, and gut-on-a-chips that absorb nutrients. This is typically what these devices look like. They're fluidic channels coupled together where you can deposit cells and have them interact strategically to produce specific biochemical interactions. And you can even string together multiple organ-on-a-chips together to create body-on-a-chip models. And these models can serve as the basis for testing out drugs and therapeutic treatments. So I am an electrical and biomedical engineer, and I'm trying to make these organ-on-a-chip models much more useful and smarter by integrating biosensors and electronics into them. Currently, the existing organ-on-a-chip models, as well as the animal models, they have the huge flaw is that you only get snapshots of what's happening into them. You set up an experiment, you can do some measurements before you start the experiment, and then after the experiment, you look at them again to see what has happened to the tissue or the live animal. But you're not getting the full biochemical picture of what's happening. You just get snapshots. So what I'm aiming to do is to integrate sensors that give you real-time and continuous data so that medical researchers can very specifically identify the biochemical mechanisms that correspond to their treatments. Yeah, for the first time, we're not just we're not just looking to see if what's happening to the tissue before at the end, where we can see exactly when a drug is starting to succeed or fail. Organ-on-a-chips are not very common right now, but in the next few decades, they're going to be ensuring that promising drugs no longer vanish, and they're also going to be accelerating drug development. Thank you.
Hello, everyone. Have you think about the food you bought last week from the grocery store? Is it on your fridge? You forget it? I think so. And if you forget it, the food is already spoiled. Hi, I'm Dr. Masood. Today, I will talk about the food waste, its effect, and how smart technology can solve the problem of this food spoilage. So let's start with the food waste. If you see the statistics, more than 4 million Canadians right now are suffering in food insecurity. And 2.3 million tons of foods are wasted. Just most of them are because of our negligence. And sometimes we forgot that we put some food in our fridge. $21 billion have been wasted just because of this food spoilage. And if you see the food banks, right now, every year, the number of people who are visiting food banks are increasing. So how can we reduce the food waste using the contemporary technologies? Oh, sorry. So let's discuss about the issues of food spoilage. First of all, if you consume the spoiled food, there will be some harmful pathogens and foodborne illnesses. As a consequence, you will not be interested to consume the spoiled food, right? And then there is some financial loss, definitely. A huge amount of money is lost just because of the food spoilage right now. And not only about the money, it's all about the resources as well. Because whenever you try to consume the foods, there are some other resources as well, which is wasted, like water, energy, and land. So if you think about the food is not the only thing which is wasted, but also the other resources. And definitely the peoples are suffering. As I have already discussed, the number of people are increasing in front of the food banks. So if you think about the food banks, here is some statistics that last year, 6% the people is increased compared to 2023. And if you think about the pre-COVID area, in that time, in 2019, 90% of increase till now. So it's the statistics until 2024. So if you think about 2025, definitely it's increasing. So the main issue is how can we detect the freshness level of the food. That's the research what currently I am doing. So some of the technologies are there. Using those technologies, we can try to calculate the freshness level of the food. So if you will go to the grocery store and take an image and try to see the freshness level of the food, if it can show you that, okay, the freshness level is 90% or 95%, you will easily buy that food, right? But if it is less than 20, you will not be interested to buy those foods. So what we can do, we can use technologies like, let's say, deep learning models, which can be integrated inside of the cameras. There can be sensors, because sometimes you can see that from outside, the food looks very fresh, but inside it's already spoiled. So not only the deep learning models or the cameras, you can also need to integrate some sort of sensors as well, so that you can get the temperature or humidity. So whenever you will collect all of this data together, then you will be able to, let's say, develop a model where you can easily see the freshness score before buying any product. Or even if you keep something on your fridge, every day you can see the freshness level as well. So the last thing is, if we can develop our own mathematical model, we have already developed our model. You can go to our website, which is crowdfeeding.ca, and you can see our results that we have successfully integrated the sensor and image data. And what we are trying to do is, like, whenever you will go to the store, buy an image, take an image, and then you can buy the food. So that's all about my presentation. If we can successfully integrate both the sensor and the deep learning models together, we will be able to reduce the food spoilage, which may improves the food security and supports the food banks right now. Thank you.
Elite sport often looks like a test of speed and strength. But some of the most decisive moments in high-level competition unfold far too quickly to be explained by physical abilities alone. For example, let's check out Connor McDavid's goal at the Four Nations Face-Off last year. And if you haven't seen this before, pay close attention to 97 in red in front of the net. The puck was on his stick for like a fraction of a second. Players are closing in around him, yet somehow he spots an opening and buries the puck in the back of the net. So this isn't just something that elite athletes do on the ice. This is actually something that we all do in our daily lives. For example, whether we're turning left at a busy intersection or we're weaving through a crowd to find our friends rushing towards a store. But how is it that we're able to attend to what matters and coordinate actions among all this visual chaos around us? Well, one idea is that we have what's called fingers of instantiation. And I like to think about these as mind fingers. It's a much of a simpler term. And so you can think about this idea that we maybe assign these mind fingers to important objects in our environment, and they stick with these objects as they move and change locations, providing us with some sort of reference to where they are. So for example, Connor here maybe has one assigned to Marner over in the corner, maybe one on the puck that he wants to track, perhaps a player in the front of the net that he wants to pass to, leaving him with one mind finger left over to pick a spot where he wants to shoot the puck. Now, researchers like myself can model and study these complex visual environments using what's called the multiple object tracking task. And I don't expect you to just take my word for it, so why don't we try it out? Your job here is to keep track of where all 4 of the spies are hiding as they scramble around on the screen. So give it your best shot. So what you probably notice is that although this is difficult, it's possible. And we find that young adults can typically track about 4 items with high accuracy. But we only have 2 eyes, so how do we do this? Well, remember those mind fingers I talked about earlier? This idea of the mind fingers is that perhaps we assign one of them to each of the items that we want to follow, and again, they stick with these items as they move. And remember, I also mentioned that these mind fingers provide location references to help us keep track of objects as they change positions. So what I did is I designed an experiment to really put these mind fingers to the test. So when you're tracking those objects, what would happen if I asked you to reach out and touch one of the items that you were already tracking? Well, you have a mind finger assigned there already, so you should easily know the location of that object. Shouldn't be too hard to touch it. But what if I instead asked you to touch one of the items that you weren't following? So for this one, you don't have a location reference for this object, so you wouldn't know where it was. This would probably be costly, and since we've already used up all of our 4 fingers, we'd have to reassign one to the object we want to touch, again, taking time, plus we're going to lose the one that we moved it away from, and so this would be especially costly. So that's what our predictions might be, but I'll show you what we actually found. So when we asked people to track objects and perform actions, in the blue bar on the left there, you can see people made very few errors when they had to touch items that they were following. But then when we asked them to reach out and touch items that they weren't trying to track, so ones that would not have mind fingers assigned to them, you can see in that green bar there, their error rates went way up, more than doubled. So it's pretty costly to do that. And we found a similar pattern of results with the reaction times. So if you look here on the graph on the right, you can see those blue bars. Participants were much faster to reach out and touch items that they were tracking compared to ones that they weren't. So again, these mind fingers were facilitating rapid action, presumably because they're sharing those locations with the motor system. So it's pretty cool. So what can you take away from all this research? But one thing you want to think about is really minding your mind fingers. If you truly only have 4 of them, you want to use them wisely. So for example, when you're driving, if you reach out and change that song, that's going to take one mind finger away from from the road where you're trying to look. Or if you're Anthony Stolars, if you cross-check a player in front of the net, that means you have one less mind finger to track the puck, which is an important job. Thank you.
Hello, everyone. Today, I'm going to be talking to you about heavy metals, their detriments, and where to find them. So, heavy metals are bad for organisms. It can cause developmental issues, it can cause short-term, long-term health implications, and reproductive health. And in fact, Health Canada has maximum permissible amounts of these heavy metals in your drinking water. And they can range from 1/1000th of a gram per liter to 1,000th of a gram per liter. And the most common instance that you may have heard of heavy metals is in the case of ocean or aquatic life. Smaller organisms are consumed by bigger organisms. Suddenly you have lead or mercury accumulating. Therefore, by the time it gets to humans, you have restrictions on how much fish you can have per week, or should. However, there is also a lot of lead, mercury, arsenic, cadmium in freshwater supplies. And they're coming from either human sources, such as mining, junkyard, landfills, or natural sources, such as erosion. And these heavy metals that are in water can cause issues. Like if you're in the city of Hamilton, there is an advisory if you're living in an old house to get your water checked. In fact, urban centers have free lead testing available for their citizens. That being said, Canada has about 7 to 9% of the world's accessible fresh water. And Canada is very restrictive about having these multi-barrier approaches in protecting lakes and rivers. However, Canada also has wells and groundwater, and those are not subject to these protections. And in fact, if you are getting your water supply from a well, you are responsible for your own water testing for things like cadmium or lead or mercury. That brings us to how do we actually test the amount of these heavy metals in your drinking water supply? You can use common testing methods such as mass spectrometry, which is the most accurate but the least accessible. And you can use something like test strips, which are the most accessible but the least accurate, because they'll just tell you if it's there or not, if it's above a certain amount. My research is all about bridging this gap between accuracy and accessibility. And accessibility coming at the cost of finances, resources, special trainings for people. So my champion in this race is an optical sensor. In the simplest terms, an optical sensor is basically just a sensor that converts an interaction of light and matter into something that can be quantified. In fact, if any of you are wearing fitness watches, you have an optical sensor on your wrist. That's what those green flashes are that you see sometimes while measuring your heart rate. In the most basic sense, there are 3 parts to an optode, or an optical sensor. First of all, you have a sampling interface. That's the part that touches the sample. You have your material exchange medium, which is the part that allows transfer of the sample to your transducer. And the transducer is the part which reacts to changes in your analyte. So in this case, if I'm talking about ion sensing, I must have a robust sampling interface that doesn't destroy on contact with water. The material exchange medium must allow only ions to pass through to the transducer. And this is my favorite part. The transducer is actually a potential sensitive dye, which quantifiably responds to a change in the analyte in one of 2 ways. Either change in brightness or change in color. So looking at the application that I have, I need a sensor that can continuously sense, because as Faizan and Sadman already mentioned, you need continuous sensing over the period of your analysis. I don't want to know if I had lead in my water yesterday. I need to know if I have it right now or 5 minutes ago when I was drinking it. It needs to be something that doesn't get destroyed by fluctuations in its environment, such as maybe you have particularly acidic water, or your water is really cold. And it needs to be specific. If I'm looking for lead, I don't want to know if there's magnesium. I want to know if it's lead in the water or not. So the thing that I've been using, it looks a bit like this. I have a wand that sticks into the water, and it has a colorful edge at the bottom. And this wire that you see are optical fibers which allow the light to travel from the light source and back into the detector. And this colorful disk at the end is basically made up of these many pixels. And each pixel is basically made of this polycarbonate pillars, which have this pink liquid, which is a polymer, which has been dyed. So there's a potential sensitive dye in there. And as you can see in the circle, there's the gray areas where the water interfaces with the polymer, and the changes can be measured against a reference dye in a sort of signal that will look like this over a period of a few hours. The idea is to make this into a pen. Thank you.
My name is Osia Patrick, and I will be discussing one of my research, which is deliberative governance and climate geoengineering, community perspective on justice-oriented frameworks. So just to start with, I want to tell you about the biggest bird in the world, which is the African ostrich. It weighs 9 feet tall, and it weighs about 340 pounds. One myth about the African ostrich is that once it faces a problem, what it does is that it hides its head and say, there's no problem here. But realize that the African ostrich is that big. Regardless of how it hides its head, you will still see it. My daughter does the same. So what she does is that when she also wants to hide from people, she closes her eyes and assumes that the problem do not exist. The same thing happens to what we do. And I want to say that climate change is real. The temperature is continuously increasing, and it is not a myth. And IPCC recorded that regardless of what we do to the greenhouse emission, temperature will continuously increase. So what do? That is where we come in. One of the interventions that has been proposed is what we refer to as geoengineering. And you ask yourself, what does geoengineering imply? It is basically the use of large machine technology to alter the Earth's environment. This is what I want you to imagine, this room. If it's cold, what do? We increase the heat, right? If it's hot, we also increase the AC. Now imagine if we could do the same thing for the entirety of the world. That is the idea around geoengineering. And basically, there are two types, the solar radiation management and the carbon dioxide removal. Why does this matter, you will ask? In the climate discourse, we tend to want to ignore geoengineering and assume that it doesn't exist. So there's a point that we say, let's not make research as relating to geoengineering. But what we are doing is applying the ostrich approach. It matters because first, the application of geoengineering affects the entirety of the world, but the vulnerability to it is felt within different communities. There's also high risks in the sense that we don't know what will happen if we try to geoengineer the world. And then there's also the potential for technocratic lock-in, which is also a challenge. And it is important for us that we look at a justice dimension to this in the sense of we asking ourselves, who bear the risks? Who bear the burden of geoengineering? Who also benefits from geoengineering? How are discussions around geoengineering happening, right? Who is involved in those discussions? What are the power dynamics that comes to play as related to this? And which are the voices that we hear? So basically, that's what I do in my research. And I apply this from a community perspective, a bottom-up policy angle, whereby individuals, communities tend to talk about their needs and how it applies to geoengineering. And we also need to conscientize the people to understand what geoengineering itself implies. And the motivation for this work is that I realize that governments are reactionary instead of proactive when it comes to policies for geoengineering and general policies in itself. And there's a need for community perspective in the work in which we do. And finally, deliberative engagement is important for us to understand the system. Thank you very much.
Hi, I'm Cam. And my current research asks what I think is a pretty important question. What would it mean to empower marginalized seniors to design the future of aging in their own neighborhoods and on their own terms? And this is a really important question because we're currently in the throes of a global aging crisis. In fact, by 2040, it's estimated that over a quarter of Canadians will be 65 years and over. Now, this seismic shift is already putting strain on health, housing, and food sectors, among so many others. Now, we're often told that, at least in part, solving this crisis means helping more older adults age in place and designing more age-inclusive communities. And though most Canadians want to age in their home and communities, the fact is they're up against a number of systemic failures, like inconsistent services, high living costs, and the rise of chronic illness, among so many others. So the problem for me is that researchers, governments, and industry leaders have pushed really quickly to try to make sure that this is solved by population-level policy and research initiatives. And I think that putting researchers and policymakers in the decision-making chair too early is going to lead to a situation where most aging-in-place strategies are going to fail to address the needs of Canada's most vulnerable communities. So I'm a community-based anthropologist. I think my work aligns with a couple of talks in this grouping of sessions this afternoon. But I am really focused on neighborhood-level solutions, where older adults and community health workers are actually the ones designing programs and services for the future of aging. What I really want to do is focus on a hyper-local rather than a population-level future of aging in Canada. Now, hyper-local places are really cool. They kind of exist within, but also exist across and between legal jurisdictions like boroughs and counties and townships and parishes. And the hyper-local places that I focus on are dense, diverse, and vulnerable high-rise communities in East Toronto. And I'm currently looking at 3: Thorncliffe Park, St. Jamestown, and Flemington Park. And the goal is to think about these as distinct sites where there are unique needs, experiences, service, and knowledge gaps. These are places that need to have a distinct future of aging. And I want to put the older adults who live in these neighborhoods, I want them to be the experts that actually drive the design of new programs, services, policies, and care environments. But how did I do that? Well, I'm an anthropologist, so I get to hang out in these communities. I immerse myself in them. I do interviews. I do community workshops, collaborative workshops. And the goal is to bring older adults, their loved ones, clinicians, community health workers, activists, and policymakers together, people who really live and work in these communities together, to actually prioritize. What are the near-term programs and services you need now? And what are the long-term policies and maybe new care environments you need in the long-term future? So I'm in the early stages of this. Ultimately, the goal is to sort of rapidly test solutions and actually develop flexible models for sharing across and between these communities. But the core thing right now is that I think, like the talk that started this grouping of 7 talks to close out the day, it's immediately clear that this hyperlocal focus is really helpful. It's not a critique of population-level research and policy, but it's a counter to it, I think a helpful complement to it. Because at this scale, we can see how geography, health, culture, politics, and economics all entangle themselves in messy ways in specific communities. At the same time, we can become very sensitive to the fact that aging in place in certain communities is very scary and isolating for people. And at the same time, we can start to appreciate why people are frustrated, angry, and feel underserved by the researchers and politicians who so often make decisions for them without much consultation. So in the end, I think what's really exciting is that we can ultimately think about helping people at the hyperlocal level to truly make a future where people age well in place. And I think ultimately it's important to study worlds within blocks before we imagine solving anything like the global future of aging crisis. Thank you very much.